Welcome to the Deep Dive. Today, we're embarking on a pretty fascinating journey deep into our DNA, but we're going beyond just the genes themselves, the ones coding for proteins.
That's right.
We're going to unpack how scientists actually pinpoint the crucial, well, control panels,
Yeah.
those hidden switches and dials dictating when and where our genes get switched on.
Exactly. Our mission here is to demystify the definitions and also the really cutting-edge methods used to identify these regulatory elements, these regions in our DNA.
Okay.
We've got some incredibly detailed source material for this deep dive. It lays out the precise techniques, the data types.
Yeah.
It's really high-stakes genetic detective work.
High stakes indeed. So if you ever wondered, say, how your body knows to make a certain protein in your liver but not in your skin, or maybe how just a tiny genetic tweak maybe miles away from a gene can still have a huge impact, well, this exploration is definitely for you.
Yeah.
We're kind of offering a shortcut to getting informed on these, well, unseen architects of gene
The
activity.
hidden machinery.
Yeah, that's exactly.
Okay, let's unpack this then. Starting maybe at the foundation. The gene blueprints. We hear about genes, but what about the parts that don't directly code for proteins? Things like introns and UTRs. Untranslated regions.
Right.
What exactly are they and why is defining them so important?
Well, introns and UTRs are really dynamic parts of the gene's message. The key thing is their definition is transcript dependent.
Transcript dependent, meaning
Meaning they can actually vary depending on which specific version of the gene's blueprint, the transcript, the cell is actually using at that moment. And this isn't just, a biological curiosity. It's actually a major challenge when we're trying to understand diseases.
how so? Why is that variability such a challenge, especially clinically?
Well, imagine two people have the exact same genetic variant, like a typo.
Okay.
But if their cells happen to use slightly different blueprints, different transcripts, that same typo could land in a totally different spot in the final message. It might have a completely different effect, or maybe no effect at all.
So for accurate interpretation, especially for diagnostics, it's absolutely critical to define these regions using consistent, well-validated, clinically relevant transcripts. We need a standard.
Right. So even if we know a gene's coding parts really well, these UTRs might still be a bit fuzzy. So what's the solution? How do scientists get that precision?
The go-to solution now is using transcripts defined by the MANE project. M-A-N-E.
A&E project. Okay.
Yeah, this project basically integrated tons of different data sets specifically to accurately define these elements. Think of it like creating a standardized master blueprint for human genes.
The consensus map.
Exactly. And it's worth noting, each gene typically gets one main select transcript that covers about 98% of genes.
Okay.
And then some genes might have extra plus clinical transcripts for, specific situations. But this standardization, it's a real game changer for consistent genetic interpretation.
Makes sense. Okay, speaking of switches, let's move to promoters. The name itself sounds pretty important, like the main on-off switch. How do scientists actually define these?
Promoters are absolutely fundamental. Yeah, they're basically the starting line for gene expression. What we look for is a specific region of open chromatin right around the transcription start site, or TSS.
Okay, open chromatin and TSS. Can you unpack those terms a little for us?
Sure. So think of our DNA like a really long thread, right? Normally, it's wound super tightly around these little protein spools called nucleosomes.
Like yarn on a spool.
Exactly. When chromatin is open, it means a section of that thread has unwound. It's physically accessible for the cell's machinery to actually read the gene.
Like opening a book to a specific page.
Perfect analogy. And the TSS.
Yeah.
That's just the precise spot on that opened page where the reading machinery begins to make the RNA message. We sometimes call this a nucleosome-free region, no spools blocking the way.
Got it. So it's the accessible starting line. How do scientists find its exact location and, well, how big is it? Is there a standard method?
There's a pretty clear two-step process. First, you have to determine the precise location of that TSS. And ideally, this should be defined in a relevant tissue or cell type, the specific cells you're interested in.
Right. Context matters.
Hugely. You can find this TSS either as the very beginning, the most 5' position of the 5' UTR of one of those MANE select transcripts we just talked about,
Yeah.
or by using a peak defined by CAGE.
CAGE. What's that?
CAGE. It stands for Cap Analysis Gene Expression. It's a lab technique that directly identifies the very beginning, the 5' cap of the mRNA molecule.
So it pins down the exact start.
Exactly. It's a very direct way to find that TSS.
Okay, so step one. Find the TSS using main E or cage in the right cells. What's step 2? Defining the promoter region itself.
Right. Once you've got that TSS pinpointed, the promoter is defined as the whole region of open chromatin surrounding it.
The accessible area.
Precisely. And this open region is typically identified using data from techniques like ATAC-seq or DINA-seq.
Or techniques.
Yeah, these are powerful methods that basically map out which parts of the entire genome are accessible or open. They give us a clear picture of those open chromatin regions.
So you overlay the ATAC-seq map with the TSS location.
You got it. And again, ideally, this data should come from epigenetic studies of a relevant tissue or cell type for whatever you're researching. What's interesting, though, is for many genes, a sort of minimal core part of the promoter seems to be consistently open across most cell types, even if the exact boundaries might shift a bit.
But what if you don't have that perfect specific tissue data? Which must happen sometimes, right? You just stuck?
No, not at all. Good point. If you don't have specific tissue data, you can still define a minimal promoter using consensus data that's available across many cell types.
Where does that come from?
It's summarized by big public resources like the ENCODE Candidate Regulatory Elements database and the Ensemble Regulatory Build. They essentially pool information from loads of studies.
Okay, so there's a fallback. Now, besides just being open, are there other molecular clues, sort of flags, that scientists look for to confirm something is really a promoter?
Absolutely. Yeah, we look for other molecular signatures. This includes specific histone modifications.
Histone modifications, those spools again.
Exactly. Think of them as chemical sticky notes on those histone spools. Certain notes, like H3K27AC and H3K4ME3, those are specific chemical tags. They strongly signal an active promoter.
Okay, a little active year science.
Pretty much. We also look for overlapping transcription factor binding sites identified by ChIP-seq.
Whoa, okay. Transcription factors and ChIP-seq. Break that down.
Sure. Transcription factors are proteins that act like molecular switches. They bind to specific DNA sequences near genes and help turn them on or off or, fine-tune their activity.
The actual regulators.
Right. And ChIP-seq, that's a sophisticated lab technique, chromatin immunoprecipitation sequencing, that lets scientists map exactly where these specific proteins like transcription factors are physically attached to the DNA across the whole genome.
So finding lots of these factor footprints in one spot is good evidence.
Very strong evidence for a promoter, yes. We also look for evidence of something called poised RNA pull 2, also found using ChIP-seq.
Poised RNA pull 2?
Yeah, RNA pull 2 is RNA polymerase II. That's the main enzyme that actually reads the gene. Poised means it's already sitting there, assembled at the gene's starting line, kind of like a runner ready in the blocks.
Ready to go?
Exactly. Indicates a gene that's ready for immediate activation. Now, this all raises an important practical question. What if you don't find this epigenetic evidence? Maybe the promoter is only active in a really specific cell type or just for a brief moment during development that hasn't been studied yet.
Right, the data might just not exist for that specific context. What's the fallback then?
In those cases where you can't confirm a region of open chromatin around the TSS with the available data, you can still define a minimum promoter region. The convention is just to take the 250 base pairs immediately upstream and 250 base pairs downstream of the TSS.
So a 500 base pair window as a default?
Exactly. It's a standard placeholder, a reasonable assumption, until maybe more specific data comes along.
Okay, that makes sense. So we've got the promoters, the immediate start sites.
Mm-hmm.
But you hinted earlier, genetics isn't always local, right? It's not just about what's right next door.
Definitely not.
What about those elements that can influence a gene from really far away? That brings us to Canada regulatory elements, or CREs. What are these, and why are they important?
CREs are, really fascinating because you can have multiple potential regulatory regions scattered around a gene, sometimes very far away on the DNA strand, yet still controlling it.
Wow.
But for any variants, any mutations within these distant regions to be interpreted clinically, you absolutely must have a known or functionally validated link connecting that specific CRE to that specific gene.
So you can't just assume because it's nearby-ish it's involved.
No way. It's like finding a light switch on, say, the other side of town. You need proof it actually controls the light in your specific building before you blame it for the light being off.
Great analogy. So this sounds like another two-step process, like promoters. First, identify the candidates, then filter for that link, right?
Precisely. Step 1, you broadly identify candidate CREs. And the clues are similar to promoters, regions of open chromatin found by ATAC-seq or DINA-seq.
Oh, okay.
They're also often marked by specific histone modifications that typically indicate active enhancer elements. Enhancers are a common type of CRE. Think of H3K27AC again, and also H3K4ME1 as common flags for these distant control regions.
H3K4ME1 for enhancers.
Okay. And just like promoters, evidence of multiple overlapping transcription factor binding sites identified by ChIP-seq is another strong indicator. Lots of activity there.
Okay, so you find these potential hotspots. Then comes the crucial second step, confirming that link to a specific gene, especially if they're far apart. How's that done?
Right, the filtering. The candidates must then be filtered down to only those that have actual experimental evidence of a direct link to the gene you care about.
And what kind of evidence counts?
Several types. One really powerful method uses chromatin interaction data. This comes from techniques like promoter capture Hi-C.
Promoter capture Hi-C, what does that show?
Hi-C techniques, in general, help us map the 3D structure of the genome inside the nucleus. Promoter capture Hi-C specifically looks for regions of DNA that are physically looping over and touching a gene's promoter, even if they're far away in the linear sequence.
So it literally shows distant regions making physical contact with the promoter.
Exactly. It's like tracing the actual communication lines, the physical wiring between that distant control tower and the specific gene it's influencing.
Cool. What other evidence works?
Another key type is functional perturbation experiments.
Perturbation, like poking it.
Essentially, yes. Scientists will directly alter or maybe even delete a part of the suspected CRE using tools like CRISPR, for example, and then they measure if that change directly affects the target gene's expression level. Does turning off the switch turn off the light?
The ultimate cause and effect test.
It really is. Also, finding one or more expression quantitative trait loci, or EQTLs, for the gene within the CRE is strong evidence.
Right, EQTLs again. Remind us.
EQTLs are specific genetic variants, maybe just a single letter change in the DNA, that are statistically linked to changes in how much of a gene is expressed. So if a variant sitting inside your candidate CRE is also known to affect the expression level of your gene of interest, that's a pretty strong clue that the CRE is regulating that gene.
Got it. So multiple lines of evidence, physical contact, functional tests, genetic links to nail down that CRE gene connection.
Exactly. You need that validation.
What's really mind-bending here, though, is the idea you mentioned earlier that the use of these enhancers and promoters isn't fixed. It can change depending on the situation. That adds a whole other layer, doesn't it?
It absolutely does. And it's such a critical point, it really bears repeating. Enhancer and promoter usage can vary dramatically across different tissues. What's active in the liver might be silent in the brain.
And temporally, too.
Yes, and also temporally, meaning across different times, like throughout development from an embryo to an adult. A crucial developmental enhancer might switch off completely later in life.
So this makes it incredibly important.
Incredibly important that when you're defining both promoters and CREs, the data sets you use, the ATAC-seq, the ChIP-seq, all of it, are derived from a relevant tissue or cell type for whatever health condition or biological process you're actually studying.
Right.
And whenever possible, also from a relevant developmental time point.
Can you give a quick example? Why could ignoring that specificity mess things up?
Oh, easily. Imagine you're studying a gene linked to a specific type of heart defect that originates during fetal development.
Okay.
If you only look at data from adult blood samples, you might completely miss a critical enhancer or promoter that's only active in developing heart muscle cells for a few weeks during gestation.
And you'd miss the relevant biology entirely.
Completely. Conversely, a regulatory element that's super active in adult liver cells is probably irrelevant if you're trying to figure out why that congenital heart defect happened. Context is absolutely everything.
Okay. Given all these data needs, the specificity required, how consistent is this whole process? Are different labs defining these regions in the same way?
That's a really important question. And, well, the reality is we have to acknowledge that defining these candidate regulatory elements using this kind of guidance, it will naturally vary somewhat. It just depends on data availability and access. Not everyone has access to fetal heart tissue data, for example.
Right, practical limitations.
Exactly. And that's a challenge because for truly consistent and, importantly, equitable variant interpretation across the whole scientific community so that a diagnosis in one place means the same thing somewhere else, these interpretable regions, these defined promoters and CREs, really need to be standardized for specific genes and diseases.
Standardized maps.
Yes. And they also need to be regularly updated as new data, new knowledge becomes available, which leads to a kind of call to action for the research community.
Okay, so what's the path forward then? What needs to happen?
Well, we really call on the community to, first, generate and make openly accessible, relevant data sets. The more high-quality, context-specific data we have out there, the better for everyone.
Sharing the data.
Crucial. And second, critically, we need to work together to produce standardized resources that define interpretable promoters and CREs across all known disease genes. It has to be a massive collaborative effort.
So pooling resources to build these shared maps.
Exactly. Collectively mapping the hidden controls of our genome, making sense of this incredibly complex system together, it's the only way to achieve that consistency we need.
It really sounds like building this multi-layered dynamic map of the genome requires some incredibly sophisticated tools. Can we maybe quickly recap the main types of data scientists are using to piece this all together?
Yeah, absolutely. It's like building that 3D city map using different survey methods.
Nope.
So we've got ATAC-seq or DNA-seq. These are your basic accessibility maps. The aerial surveys showing the regions of open chromatin, essential for finding both potential promoters and CREs. Where can the machinery actually land?
Got it. Open areas.
Then CAGE, cap analysis gene expression. This pins down the 5' cap of mRNA. It's vital for defining the exact start of promoters and the 5' UTRs. It tells you precisely where transcription begins, and it also highlights if there are alternative transcripts starting elsewhere.
The starting point marker.
Yep. Then the histone modifications, like H3K4ME1, H3K4ME3, H3K27AC. These are the chemical flags on the DNA spools. H3K4ME1 often near enhancers, CREs, H3K4ME3 and H3K27AC near active promoters and other regulatory elements. They signal the activity state on, off, ready.
Activity flags.
That's right. ChIP-seq is our zoom lens for specific proteins. It finds transcription factor binding sites and poised RNA pull 2, showing exactly where those key regulatory proteins are sitting or where the reading machinery is waiting.
Protein locations.
Then, promoter capture Hi-C. This traces the physical connections, linking distant enhancer regions to promoters of target genes. It shows who's talking to whom in 3D space. Though you have to be a bit careful, some interactions might be transient and not functionally essential.
Communication lines.
Exactly. EQTLs, expression quantitative trait loci. These link specific DNA variants directly to changes in gene expression. They're key for identifying functional promoters and CREs by showing a direct consequence of variation in that region.
Genetic links to activity levels.
And finally, experimental perturbation. This is the direct functional test, altering or deleting an element to see the impact on gene expression. It's the gold standard for cause and effect, though you always need to consider if the lab assay is truly representative of the endogenous situation inside a cell or organism.
The functional proof. Wow, that's quite a toolkit.
It is, and remember, layered over all of this is that crucial consideration: tissue specificity and temporal specificity. The data absolutely has to fit the biological context you're asking about. No single tool gives the whole picture, but used together, they build an incredibly detailed understanding.
What an incredible deep dive that was. We've really journeyed from, the basics of introns and UTRs, just understanding why defining them accurately matters so much, all the way to the sophisticated world of promoters and these candidate regulatory elements, the CREs.
Yeah.
We've explored the really precise methods, the complex data types scientists are wrestling with to map these vital genetic control panels, understanding not just what they are, but how they're found, and crucially, why it's so important.
And understanding these regulatory elements really is fundamental. It's not an exaggeration to say they dictate the precise choreography of gene expression. They're the conductors of our biological orchestra.
I like that.
And this knowledge, it's not just academic, right? It has truly profound implications for understanding health, disease, and especially for developing the next generation of targeted therapies that might go beyond just the protein coding bits.
Right. So what does this all mean for you listening right now? Well, consider this. How might precisely targeting these subtle, often distant regulatory regions in addition to the genes themselves, how could that revolutionize future treatments? What new discoveries, what new frontiers, and maybe personalized medicine might emerge as the scientific community keeps collaborating, keeps opening up these crucial genetic maps, making these invisible controls of our biology, well, visible and understandable for everyone? Something to think about.